Now I talk about part 2, radioimmunoimaging. We call it RII. First principle is a specific monoclonal antibodies are labeled with radionuclide. After being introduced into the body, they can be combined with the tumor cell-related antigens directly and specifically. After a period of time, the radioactivity of tumor site accumulates to a certain concentration. The size, the location, and the range of tumor and metastasis can be displayed by a planar or tomographic imaging with gamma camera, SPECT, and PET. Advantages of tumor radioimmunoimaging. First, the accuracy and sensitivity of tumor localization in vivo are 90% and even 100%. Second, in vivo, 0.5 centimeter lesions can be detected, and the metastasis of lung and brain can be detected. The half-life of antibody in blood can be observed for 6 to 9 days after tumor size. The process of radioimaging. First, we should select a monoclonal antibody, radiolabeled with it. And then we introduce this probe into the body, and within the body, binding to the corresponding antigen. This antigen usually expressed on the tumor or a specific tissue surface. And then we can use SPECT or PET to do the tumor imaging outside the body. Then we can know the size, the locations, and the range of the tumor and the metastasis. Requirements for target antibodies. First, should have the high specificity and stability, and then need high specificity after labeled. That means you labeled antibody within the radioisotope, it cannot change its specificity. And the third, fast non-target tissue removal, so it shows low background. And the last should have the high tumor tissue concentration and long accumulation time within the tumor. And this is for antibody. And second, commonly used radioisotope for SPECT, SPECT, we usually use iodine-131 and technetium-99M and indium-111 to label. And for PET machine, we usually use fluorine-18 labeled, and sometimes carbon-11 or copper-64. To date, 5 technetium-99M labeled or indium-111 labeled murine monoclonal antibody have been approved by the FDA for single photon imaging computed tomography imaging. It's for SPECT imaging. Among these, radiolabeled antibody to tumor-associated glycoprotein 72, we call the TAG72, prostate-specific membrane antigen, we call the PSMA specific, and CEA, carcinoembryonic antigen, and epicell adhering molecule have been approved by the FDA for cancer imaging. Now I introduce them in detail. First, indium-111 B72.3. So this is the mouse-derived monoclonal antibody. Has a high expression in colorectal adenocarcinoma and ovarian epithelial cell carcinoma. Can also be used in breast cancer, small cell lung cancer, pancreatic cancer, gastric cancer, and esophageal cancer. and 2 tetramethyl-99M labeled CEA-FAB. CEA-FAB is an anti-CEA mouse monoclonal antibody fragments. Has an advantage of small molecule weight, strong penetration, and fast blood clearance. It has great value in detecting primary and metastatic colorectal cancer inside or outside the liver, as well as lymph metastasis of breast cancer. The third tetramethyl-99M labeled NRLU10-FAB. It is an anti-lung cancer antibody fragment, which is highly expressed in small cell or non-small cell lung cancer. It is of great value in the classification of lung cancer stage, detection of metastasis, differentiation of benign and malignant tumor. And the last indium 111 labeled capromab. It is a mouse-derived IgG antibody which can combine with antigens in the cytoplasm and in the cytoplasm of prostate cancer cells. It has great value in detecting pelvic lymph metastasis of prostate cancer. See this picture. The upper row is CT scanning. The lower part is CA scanning. The left shows no primary metastasis. The right shows the local reference after operation. And see this picture. This is 44 years old female, 10 months after resection of right colon cancer. Recently, detection found serum CEA increased and do the PET F18 FAD scanning negative and do CEA radioimmunoimaging positive. So by operation, the right pelvic metastasis was confirmed. And this picture shows radioimmunoimaging is specific to the CEA antigen. And also see this picture. The upper row shows the tumor imaging of antibody imaging. Tumor imaged by the antibody imaging in mice with a Cooper 64 labeled antibody. You see these two tumor-bearing mice, and the left one and the right one both showed clear for tumor. And the lower part is a home lab pictures with monitoring allograft rejection and in vivo with iodine 125 labeled anti-tau5 monoclonal antibody injection. We do the forceful autoradiography. You can see from 1 hour, 12 hour, 24 hour, 48 hour, and 72 hour. You can see the rejected graft clear. For radioimmunoimaging, these monoclonal antibodies are mainly used for stating this disease in patients suspected of recurrent or metastatic disease. However, their overall clinical impact has been limited. Advances in protein engineering have facilitated the construction of antibody-based molecules that retain binding characteristics of intact monoclonal antibody, while optimized for in vivo diagnostic purpose. Just shown as this picture, the antigen binding specificity of a monoclonal antibody is dictated by the 6 complementary determining regions, we call CDR, that are found in its variable domains. 3 in both heavy chain and light chain, we call VHVL. Molecular engineering techniques have facilitated the construct of single chain FV, we call SCFV molecules, that are comprised of the VH and VL domains of a monoclonal antibody joined together by a short peptide linker. The scFv is about 25 kD and can be readily produced in a wide range of express systems. Most importantly, it recapitulated the binding specificity of parental monoclonal antibody. scFv molecules represent the basic building blocks of antibody engineering and can be used to construct a wide range of molecular structures that vary in their size and valence. So we can get a human, mouse, chimeric antibody, smaller molecule, high specific scFv binding antigen, peptide fragments of synthetic hypervariable region sequence, special modified antibody. Our ultimate goal to improve specificity, reduce molecular weight, and reduce allergy to human body. Okay, that's all for this part.